I'm Karin High. I'm a professor and vice chair for research at anesthesiology at UNM Health Science Center. And it's not advancing. There's my team. We have an antibody for chronic pain management that was generated by Dr. Kunamneni. And it's very similar to vaccines that he's developed with Ravi Devasala. They're now at Mayo Clinic. And we have validated the antibody for chronic pain in animal models. I should say Marina Montero, the lab manager, has done that. We had funding, a little bit more remaining from NIH R01, and we've just received a DoD to follow up this work and get this humanized and in a more commercial ready state. So what is the innovation? It is a single chain fragment variable, or ScFv antibody, for non-opioid treatment of chronic pain. You probably can't see my graphic if you have the people along the side, but if you would move that somewhere else, the monoclonal antibody is a Y-shaped structure. And what Dr. Kunamneni has done was to take this small arm, and these small antibodies are called single chain fragment variable antibodies. And he has engineered them, and we have tested them. These are known to have a binding activity like the monoclonal antibodies, but with stronger binding affinity, increased tissue permeability, and the small ScFvs are brain and nervous tissue penetrant due to their smaller size, which is what is needed for pain therapy. Now, what Marina Montero has found is that a single dose permanently reverses chronic pain in 3 different animal models, and it prevents anxiety and depression-like behaviors. We know that more than 20% of US adults have chronic pain, which is more than 40 million Americans right now. More than 3 million new chronic cases develop each year. 80% have high impact pain. What this is chronic pain that will continue on, and it will very likely become a cause of long-term disability. There are currently no effective treatments for chronic pain, and chronic pain cannot be cured. In the United States, non-addictive replacements for opiates are a high priority. Chronic pain is linked, as I said, to anxiety, depression, and reduced quality of life, which leads to disability and potentially opioid dependency and drug overdose deaths. In 2015, the CDC produced this map of the United States, and you can see that New Mexico was unfortunately highest among drug overdose deaths. In 2018, we have reduced this number, but we still appear in the dark, awful designation. Now, there are more than 67,000 drug overdose deaths in 2018 in the CDC's tally, and it involved opioids almost 70% of the time. So pain is costing an estimated $560 billion annually in direct medical costs, low productivity, and disability programs. The US's most prescribed pain drug is a $6.3 billion market. The ScFv antibodies fall into the biological neurological drug market. Now, while FDA-approved monoclonal antibodies are used for arthritis and migraine headaches and are not very nervous tissue penetrable, the ScFvs have been used for more than 10 years as cancer therapy, so that it is well-developed state of treatment. Now, these are the studies that Marina has shown. We do these chronic models. In this case, I'll show you the oral facial pain model that she and I developed. And while normal animals maintain a steady level of sensitivity to pokes on their face, the animals with an orophacial pain model have increased sensitivity until she gave them an intraperitoneal single dose treatment. And those animals with 2 different P2X4 ScFvs returned to naive baseline in the next week and were maintained there permanently. Whereas the animals that maintained the model without treatment remained at this hypersensitive state permanently. Also, we found that, like the naive animals, the treated animals maintained anxiety-less states. So the ScFv prevents the anxiety and also prevents many of the depression measures like the naive state. In a more recent chemotherapy-induced pain model, she gave oxaliplatin, much like the clinical treatment scheme, on and off 3 times over 3 weeks. And when giving the antibody treatment alongside the oxaliplatin treated drugs with untreated antibody had this very hypersensitive state and cold on their feet, cold hypersensitivity and mechanical hypersensitivity, whereas the treated mice were returning to naive state, and within the treatment time recovery, we're back to baseline, which was very startling, and there is no other treatment like this available. If you were to stain nerve cells with the commercial antibody to observe the P2X4 that is on the nerve cells, this is what it would look like in a dish, thanks to Dr. Sabrina McElrath at the VA. But if you pre-block the nervous cells with the antibody, then almost no stain is apparent associated with the nerve cells. With the Western blot, this is a control His tag that we applied because the antibody has a His tag marker. And when we took a brain stem homogenate, 4 animals also had the His tag. So this is a demonstration, the first preliminary demonstration, that the antibody does penetrate into the region of the brainstem where the orofacial trigeminal nerve innervates the teeth and the orofacial structure. The nerves on the other end that enter the brainstem were labeling in this brainstem area. And probably at least 5% of the antibody that was given as a single dose into the abdomen was still apparent in the brainstem. Now, Dr. Kunimneni has gone on to further these steps towards commercialization, and he's done computer modeling. This turquoise ribbon structure is our antibody, and using a peptide with the human sequence to the P2X4, he has shown that it will bind, and when he uses it in a cell receptor modeling program, it totally enables the actions of the P2X4 ion channel, and so that it can no longer function. He is also generating humanized variants of the VH and VL, the short arm IgG, now as a ScFv, the small antibody. He's humanizing it for potential human use and commercialization. And he is also, with this product, we'll be able to test the humanist score, probably with the CRO that we're about to work with. So as a new part of the project, Aaliyah Goins, who's just been accepted to the UNM Biomedical Graduate Student Program, and Dr. Sasha Alls have been working with a CRO, and they have a human validation platform. And in this case, human blood is transformed into neuronal-like cells over time with their growth factor treatments. And Alea is able to stick the cells and get recordings. And over time, the cells begin to have neuronal-like properties with which we can test after we activate them. Then we can see if we can prevent the activation with the ScFv antibodies. So the current status is that these proof of concept studies and data all provide translational potential. And we're beginning to see that we're very likely to have a human crossover and not just with mice to block the chronic pain. With the DoD, the NIH, and some VA merit grant funds, it's going to allow us to work with a commercial research organization and to humanize, validate, and upscale the product. So with the new team members, Dr. Sasha Alas, he will be able to look at the human neuronal-like cells and validate their ability to block pain-like responses. And with Dr. Stephen Bradford, also at UNM, we'll be able to look at immune cell interactions because immune responses are also very prominent in pain. We have been working with the UNM Rainforest Innovations to align with investors, and this work will continue. But we have a great hope that this is a non-opioid solution for the opioid crisis, and that the ScFv Immunotherapeutics will be one of the first effective chronic pain management tools. Thank you.
Thank you, Karin. Over to our panel for questions.
Hey, Karin, this is Mike. Really liked your presentation. Congratulations on the DoD award in particular. How far do you think you can take the non-dilutive funding through the commercialization process until you need outside funding?
Well, we can have outside funding if we can just get, we have a potential investor, but it will require funding with them. And in that case, then we could hopefully get $500,000 funding to bring this forward, because I believe that we're virtually there. Once we get these in the lab human assessments, I think we're ready for the commercialization.
And have you brought a regulatory consultant in to give you advice on FDA pathway?
Not yet. I have a consultant who has just brought a commercial antibody for sale to a company that's taking it to FDA. And if we can just align one more grant with them, we can potentially move forward.
Okay.
Do you have a sense of how much money you'll need for your first round of investment, and what kind of milestones are you looking at for that first round?
Well, with the DoD, we have the milestone within less than a year now to get this product humanized and tested in the animals. And then the next year would be the next milestone. The second year will be to see how this is functioning in the human neuronal-like cells. Then from there, I'm hoping that the investor who has just taken their antibody to market will be able to fill in all the knowledge gaps that I would have for the commercialization and to get this to the clinic down the pike. We do have clinicians that are very aware of this product in our own department, and they have been very encouraging.
Hey, Karin, this is Mike again. So as you think about an outside investment, have you thought through some basic terms at this point? Did you want to do convertible note? Is this price round? Does those terms all make sense to you? I mean, have you thought about how you might structure the investment?
No, sir, I have not. The investor consultant is expert with that, and he is educating me along the way. I've worked with the New Mexico State Ascend program, and I'm learning, but no, at this point, I've been a basic scientist for over 30 years, and my knowledge in that area is very limited, but the UNM Rainforest Innovations has handled a great deal of that, and they're very able advisors.
I totally agree. I just wondered if you've got how far down the path you've gotten. So sounds like you're definitely doing the right things.
Thank you. It's good to know.